Thank you everyone for coming today to the joint GlycoNet ACS Carbohydrate Division webinar series. My name is Warren Wakarchuk and I'm the Scientific Director of GlycoNet. And it's my great pleasure today to introduce our first speaker, who is Professor Sabine Flitsch. And Sabine was trained as a chemist first at the University of Munster in Germany, and went on to get a PhD from the University of Oxford. She then spent some time in the US at MIT with Gobind Khurana before returning to the UK to hold academic positions in a few places. But she is currently a professor of biological chemistry at the University of Manchester, where I actually got a chance to meet Sabine a couple of years ago at a workshop there. And I must say I'm looking very, very much forward to her presentation today. So Sabine, the floor is yours.
Thank you very much. Well, first of all, many thanks to GlycoNet and also the ACS division for inviting me. So it's a great pleasure to talk a little bit about our recent work. And I've given it a very generic name, synthetic and analytical tools for glycobiology. What I'll be talking mainly about is enzymatic synthetic tools and some sort of companion analysis we have developed with these, by necessity, I should say. So I'll start off with this sort of slightly older work, just to say a little bit about enzymatic synthesis. So we're very interested in using enzymes in organic synthesis in general, but particularly for carbohydrates because they're such challenging targets. So the structure 5 you can see here is a glycopeptide. It's an O-mannose glycopeptide, part of this alpha-dystroglycan complex, very important in muscle function. Chemically, this is a very challenging target. In particular, if you want to devise a synthesis that gives you very great flexibility in terms of peptide sequence. So if you want to make a whole lot of peptides with the same glycan structure, that's pretty challenging. However, if you use the enzymes, the glycosyltransferases that are involved actually in the biosynthesis of these, then you can actually very quickly assemble this complex tetrasaccharide in 3 steps using just the GlycoNet transferase, a GAL transferase, and a silaltransferase. That is very easy, and you can actually do it in 1 step. So you can just throw everything into one pot, start with a chemically synthesized glycopeptide 2, and then get a structure 5 with very, very high selectivity. Because we were using enzymes that were not necessarily biosynthetically responsible for this target, we felt we needed to fully characterize it by NMR, and I've missed out his name here. This is Göran Wittmann, who helped us with that. And we could really show that with minimum purification, we can get highly pure tetrasaccharide as shown. So these enzymatic reactions are obviously extremely useful. And if I could sort of summarize the state of the art a little bit, is that these enzymes are really very easy to use. You just have to sort of mix them up with a substrate and incubate them. The reaction conditions, as you all know, are very mild, and the selectivities generally are very, very good, so that it requires minimal purification. So we think that enzymes are extremely useful to what we might call democratize glycan synthesis, because the idea would be if you could make these enzymes all available, in particular to biochemists and biologists who may be not so familiar with synthetic strategies, then there would be fantastic tools and give people access to glycans and also glycoconjugates like these peptides I'd shown you before. So what are the challenges in the field is? So the challenges we think are, first of all, people need to sort of really know about these biocatalysts. At the moment, you need to go through the literature or go to databases. The Casey database is a fantastic database, although it's more sort of structural based. You need to get access to these biocatalysts. And also, we should admit we still have a very limited range of these enzymes, of these biocatalysts. So not every structure is really available to people who want to use them. And then I think what is also very challenging is sort of really the analytics here, because they're not straightforward. So I want to talk today a little bit about how we might expand the range of biocatalytic activities which we have currently, and also talk a little bit about how we measure these expanded activities, and talk a little bit about some analytics we have looked at. So if you generally want to have more diverse enzyme activity in this, what we call carbohydrate active enzyme toolbox, and I should refer to this fantastic database which we all have and we all use, the KZ database, then we have a whole range of different techniques we can use. We can use new enzymes activity. If you've seen a natural product, a particular polysaccharide or so, we could go after the biosynthetic enzymes, which need to assemble these. We can use directed evolution these days, although we do need to start often with some sort of activity. There's rational redesign, which has worked in some cases. You can look at metagenomic databases, looking for homologs of existing enzymes. And maybe more futuristically, you can look at de novo design. So that's in its infancy, but hopefully in the future, we'll be able to do that. But what I want to focus today about is promiscuous activity of existing enzymes, which we have classified, but which can give us actually some useful biocatalysts. So that's all we're really after. We want to do in vitro synthesis of these carbohydrates, and we want to find a catalyst that can catalyze a specific reaction. We're not too worried whether this is a native activity or whether it's maybe a promiscuous activity. So that's sort of the topic today, looking for promiscuous enzyme of activity of existing enzymes. So promiscuous activity of enzymes is actually a very old topic. People have thought about this for a long time, and it's now believed that actually most enzymes have some promiscuous activity. You all know that enzymes normally are highly selective in a native biological context. They would pick a particular substrate out of a range of substrates and then form this complex and make a particular product. So they're highly selective. We know them as highly selective catalysts. But actually, a lot of enzymes have, if you look carefully, some promiscuous activity. And that's obviously very important for evolution. And as I said, there's some beautiful papers that have been published, so like these here, where people discuss the role of promiscuous activity in evolution. The only thing you need to do is very often is you need to use much more enzymes, because the catalytic activity is not at all as great. And sometimes we're talking about, significantly lower activity. And you have to have quite good analytics to look for this promiscuous activity. But what I hope I can convince you is that nevertheless, even natural enzymes can really come up with some very useful activities. And I'll give you some examples of these. And what sort of reactions am I going to talk about? Of course, I'm going to talk about glycosylation reactions, which are the most challenging in this field. But start with actually oxidation enzymes. So these take hydroxyl groups in glycoconjugates and oxidize them. And to talk a little bit about acylations, if I've got time. So let me just talk about oxidations first. Here, our sort of enzymes to start with is an enzyme called galactose oxidase. Again, it's a very old enzyme. It's got a copper center. The mechanism is quite well known. It's got a very shallow active site. And the beauty about it is that it can really tolerate huge groups here. You can oxidize galactose, for which is this highly selective, on cell surfaces, on proteins, on glycoconjugates, and so on. And it would always selectively give you the C6 oxidized product. Sometimes the acid, sometimes it can over-oxidize. And the beauty is also it just uses molecular oxygen. The disadvantage of the native enzyme is that it is actually not very promiscuous. It really only oxidizes galactose or galactoside, and also actually talose, I should say. But it's really been mainly used for galactose. So here what we did is use actually directed evolution. And I should say, actually, this is, I haven't really put the paper in here, this is really very much based on Frances Arnold's work, first of all in terms of her developing a directed evolution, but she's actually worked on this enzyme as well and generated some mutants, which were extremely good starting points for generating more promiscuous activity. And I don't want to go through details of directed evolution. You probably all know about it. You make random libraries and then you just select for activity. And it turns out now we've got a whole set of mutants which have some really interesting activity. Of course, what they do is they take sugars and, for example, we have a mutant that can actually oxidize now mannose on cell surfaces. So these are yeast cells. We can selectively introduce the aldehyde group. And once you have the aldehyde group, it's not a particularly stable group, but if you generate it in situ, you can do some very nice bioorthogonal click chemistry. For example, click on some fluorescent groups. So here you can see this lighting up these yeast cells because we've oxidized cell surface mannose residues. We've also used it on DNA libraries, so completely sort of unnatural constructs in that sense. And also we've used it for glycoproteins. You can do it, of course, it has been used for a long time for glycoproteins to oxidize galactose, but we have found that some of the mutations, and this is a mutant that is a sort of workhorse in our lab, this mutant F2, it has several mutations compared to the wild type, can actually, interesting, very selectively oxidize N-glycosylaneurominic acid on the trisaccharide, the acid itself, but also on glycoproteins. And what it does is, interestingly, it goes for that extra hydroxyl group that glycolyl, sorry, my pointer is going. This extra hydroxyl group N-glycosylaneurominic acid has in red here, which sialic acids obviously don't have. So with this enzyme, you can actually distinguish between sialic acid and glycosylaneurominic acids. Only the sialic acids, the glycosylaneurominic acids get oxidized to the aldehyde and can be labeled. And you can show this by just cleaving all the sialic acids off with generic siladase and then just show really by HPLC that it really only is the glycosylaneurominic acid that's been oxidized. So that's a really nice tool to selectively oxidize these. And actually, we don't even have to have sugars as such. We had a very nice collaboration with Bruno Linklau and show you a little bit more of the work with him, making a whole range of deoxyfluoro sugars. So this is 3-deoxy-3-fluoro-fucose. We wanted to use them as building blocks to incorporate them into oligosaccharides. We've got an ongoing project with him. And he was making these and trying to make them chemically, but we're really struggling with this last oxidation here of this compound here, which using all the known oxidants he tried, gave him all sorts of decomposition products and so on. And what we found is that our galactose oxidase F2 mutant, which has really much broadened activity, actually can very selectively oxidize that primary hydroxyl group in the presence of all of those, and then it cyclizes, and this reaction enzymatically is very clean. So we've now really generated a much more promiscuous enzyme, albeit in this case through directed evolution. So this is a really beautiful enzyme system. And we're really trying to, at the moment, make that more widely available. We've stabilized it by putting it onto solid support. And we've also used it in flow on gram scale. So this is really a very, very nice oxidation catalyst, useful for certain cell surface and protein sugars, but also for small molecules. Quite a universal oxidation catalyst using just molecular oxygen. So that's sort of the oxidases. But as I said, really what people are obviously always interested in is glycosylation. So in an ideal world, we want a toolbox that generates any glycoside we want with any selectivity we want. And of course, we're very, very far away from that. We have a good toolbox, but it's really, I think you'd all agree, is extremely limited. So one question you might want to ask is, okay, we can make natural sugars and natural glycosides. What about unnatural glycosides? And as I said before, we're very interested in introducing fluorine into different positions of glycosides. These are very, very useful for NMR studies. If you look at binding of sugars onto proteins, you can do fluorine NMR, but they're also very useful to really probe substrate specificity. So we think a toolbox of fluorinated sugars would be extremely useful to really understand carbohydrate interactions. The monosaccharides are challenging to make already, but actually then to use them and make glycosides is even more challenging. So we're trying to find enzymatic methods to do this. And in the first instance, we would just ask the questions, can we just use natural enzymes that work on, let's say galactose or glucose and do the glycosylation? Do they tolerate these deoxyfluoro substitutions? And not only one, but in some cases we want to introduce two or even more. And so in this particular case, we've now got, it's not a sugar really anymore, it's got a tetrafluorinated galactoside, you could say. One of the glycosylation reactions that is extremely important in nature are phosphorylations. So we got hold of a whole lot of different anomeric sugar kinases. And I should say almost all the enzymes I work with here, we are working with, are expressed in E. coli. So we are very keen to develop enzymes that are very easily accessible and can be very easily expressed in E. coli. And in fact, we often don't do that ourselves. We do this with a company called Prosomics who does it for us. And you can see actually these enzymes are actually remarkably tolerant for these fluorine substitutions. So all of the panel, all the structures I show here on the right can be actually phosphorylated with one of these 13 enzymes. I'm not saying every enzyme can tolerate every substrate, but if you have a panel of enzymes, and as I said, if you're also able to put excess of enzyme, and in some cases we really had to put quite a lot of enzyme in because the promiscuous activity was not particularly strong, you can really get on preparative scale these glycosylation reactions to work. What we're having here is starting to have a little bit of a problem of screening. We've got 13 enzymes, we've got all these different substrates. And this is when I would start talking a little bit about analytics. So in this particular case, we are really very keen to implement mass spec-based techniques where we basically just take crude incubation mixtures. Shown here, sort of coming out of our reaction cycle, spot them onto a plate without any further purification, and then directly analyze them by mass spectrometry. And one method that is particularly nice is this DAISY method, which is a desorption method. So this is under ambient condition. You don't actually need vacuum here. You put the solvent stream onto here with a mass inlet. And then you can directly interrogate the surface for particular mass ions. You can visualize that surface then in a sort of imaging techniques here by just asking what m over z value is observed here. And what you can see here with the matrix is that on the left-hand side you have all the sugars, on the top you've got all the enzymes, on the right-hand side you've got the mass you want to measure, and you can see that in a lot of these combinations, different lights, spots light up. So these are the spots of your crude incubation mixtures and show you that you get some positive results, and you can then follow them up with other analytics. And this is a very, very fast way, and it's a label-free way, so it doesn't really rely on us looking at a particular fluorogenic substrate, for example, and it's not quite as fast as a fluorescent substrate, but you can interrogate this plate with about 45 seconds per sample. So this is a really, really fast technique, not requiring any analytical time. So this is a really nice way of looking for this sort of promiscuous activity of unnatural substrates, because you don't rely on assays that involve natural substrates. And this theme of fluorination, now we've made glycosylphosphates. Now we've made the building blocks, and as I said, this is all done by Bruno Linklau. We can now incorporate them into glycosyltransferase reactions. And again, we find that not every substrate is tolerated, and particularly in the 2 position, you really struggle with glycosylation for obvious reasons. But actually, in other positions, you really can make quite a lot of different varies fluorinated oligosaccharides. So this is an example where we start with this 3-fluoro-3-deoxy glucose, fucose, or with fucose itself. We activate it to make GDP fucose with this one enzyme system, and then add acceptors, LACNAC acceptors, and 2 fucosyltransferases, and you can make these sort of Lewis structures here, which are selectively fucosylated. And as I said, they will be very useful for further structural studies. So that's all very nice. So this is now deoxyfluoro sugars. So they seem to be tolerated, and there seems to be a lot of scope to use these sort of unnatural structures. What about looking at enzymes that have a known selectivity? So in this case here, I'm talking about silaltransferases, which I've shown some examples of before, where you can just take CMP sialic acid and transfer it onto a substrate, and the silaltransferase will determine which linkage you're making. We all know about these silaltransferases. Question is, can you use these enzymes for other natural sugars? And this is a collaboration with Martin Fassioni here at York University, who was very interested to make glycosides that contain pseudominic acid, which is a bacterial sugar, and it's quite distinct from sialic acid. It's got, oops, my pointer does not work terribly well here. So in the gray box, you can see the structure of this pseudominic acid, and those of you familiar with sialic acid particularly recognize that the side chain is quite different to that of sialic acids. And what we find is when we screen the whole panel of silaltransferases, which we had in the group, and again, these are bacterial silaltransferases, we can actually identify 4 that were actually able to take the CMP pseudominic acid donor as a substrate rather than CMP sialic acid, and in particular this Pesterola multicida silaltransferase here, which is a slightly truncated version, was actually very, very good at achieving this glycosylation with reasonable yields on monosaccharides, and in particularly good on disaccharides, as you can see. So you can actually use natural transferases to actually transfer sugars other than their natural donor substrate. And we've now shown this with sort of several examples. Another class of enzymes which are extremely useful in making oligosaccharides and have been widely used are these beta-1,4 galactosaltransferases. So these would make LACNAC and lactose and so forth. And again, we've looked at some of these bacterial enzymes, for example, this LGTB enzyme, which normally takes here UDP galactose in yellow and adds it onto glucose as an acceptor substrate to make these lactose and LACNAC derivatives. And as I said, this has been widely used by scientists. The question was, would these enzymes also accept UDP glucose as a donor substrate, as a promiscuous activity? And we were particularly interested in that because once you do that, you can actually use this as a polymerase to make cellulosic type fragments. Because the product is an acceptor substrate again, and you can go around and cycle and just make a whole line of batteries. So we screened quite a lot of these enzymes, and we found actually, as I said, there's this LTGB enzyme could catalyze this reaction. And I should say this is an extremely slow reaction. We need to leave this for several days on end. But nevertheless, the products are actually quite useful products. In particular, if you can tag them. So we work with Carmen Galan, who has I-tagged the acceptor substrates for us to make I-tagged glucose, which also aids, I should say, analysis, because you can then just mold all of the substrates, products, and so forth. And then we can actually further elaborate that with the same enzymes actually by adding UDP galactose or the oxidized version of UDP galactose and tagging it at the end. And then you have sort of bifunctionally tagged cellulosic fragments, which you can then use as enzyme substrates, for example, for LPMOs or endoglycosidases and so on. And as I said, all these different reactions you can monitor directly by MALDI-TOF because the I tags are such good. They're basically mass spec tags. So really, if you look very hard, but as I said, I should stress this is quite a low activity. You really can get some sort of very unusual and unnatural activity. And in fact, it's often the same enzymes that have this much broader substrate specificity. So we've used the same enzyme here in a slightly different context. What we were very interested in is make building blocks for an automated glycan assembly, because it turns actually out enzymatic reactions have this sort of reputation that you can only do them in small scale. But in industry, biocatalysis is really used to do reactions on large scale because they're often the single reactions. You get this high selectivity. Enzymatic methods, biocatalysis is actually quite useful for scale up. So we challenged ourselves to make a building block for automated glycan assembly, which, as you all know, eats a lot of substrate, by enzymatic methods. And the target was this LACNAC building block, which is very, very common in many saccharides. And for this particular case, it was a cell surface polysaccharide. This tetrasaccharide, which I just shown on the bottom right. And then the question was, of course, that after the enzymatic assembly, we'll need to activate it and protect it. And in particular, what you really need for these building blocks is a 2-trichloroacetate protecting group, because otherwise you've got problems with your coupling reactions, and obviously for activation, things like, for example, a thioglycoside. So the question was, substrate 1, is that actually a substrate for our galactosaltransferase, which normally just accepts an acetylglucosamine? And it turned out for this particular enzyme, it was, and it catalyzed this reaction in quite good yield. So it was actually quite tolerant of this 2 substitution. The other challenge was really to do this in gram scale, and the core substrate for these transferases, as you all know, is UDP galactose, which is fairly expensive. So you don't really want to do this reaction using gram scale UDP galactose. It would make it extremely expensive. Normally what you would do is you would sort of regenerate the system. So there's a very beautiful, well-established system, 3-enzyme system, regenerating UDP galactose from sucrose, making UDP glucose and then UDP galactose. So you need the SUSI enzyme and you need the epimerase, which sometimes can actually cause problems because you get transfer of UDP glucose, as we just shown before. And what we found is that alternatively, you could also use lactose as a donor here. And lactose is obviously super cheap. It's a waste product. And the enzyme actually catalyzes the transfer of galactose from lactose onto this acceptor substrate 1 in the presence of UDP, just generating glucose. This is obviously an equilibrium, so you need to add huge excess of lactose to drive this reaction. But lactose is very easily separated by recrystallization actually at the end. So really, Andrea Marchesi, who's a PhD student, very talented PhD student in my group, actually managed to produce 2 in very, very good scale, sort of just in one step. We then managed to put that into the GlycoNet system. This was a collaboration with GlycoUniverse, and could show that we can indeed use this enzymatically assembled building block for chemical synthesis. And actually, we find that quite a lot of enzymes, not all, but a lot of enzymes, could tolerate galactose and glucose together. So another enzyme is this mucin GalNAc T2 transferase, which is involved in the first glycosylation step in mucin biosynthesis, or is one of the enzymes of the many enzymes in humans. And this T2 variant in particular, we could also, and again, the kinetics are orders of magnitude less than that for UDP galactose. But nevertheless, by adding a good amount of enzymes, we could actually preparatively produce the GlycNAc derivative rather than the GalNAc derivative. So in vitro, you could really push this reaction to push this enzyme to accept UDP GlycNAc. And here we started to hit another sort of analytical problem, is how do you actually prove that you've made the right compound? Because as you're all aware, we have this epimerase, which could be anywhere, and any activity we might see by mass spectrometry could be due to the fact that we've got epimerase in there that converts UDP GlycNAc to GalNAc, and then you get the transfer of that. So we were very, very keen to show that we actually were making the GlycNAc derivatives and not the GalNAc derivatives. And here we sort of almost had to develop a hyphenated mass spec method, and I really put credit there to my colleague Claire Ayers, who was our mass spectrometrist at that time, or colleague in mass spectrometry, and she introduced us to ion mobility spectroscopy. So this you can couple with mass spectrometry. So you put your samples into the mass spectrometer, you fragment them, and then you put the fragments after fragmentation, for example with CID, through this ion mobility tube. And then you just look for differences. And what we could observe is that even though the differences between the glucose and the galactose are extremely small, ion mobility was able to very reproducibly separate the two so that you can unambiguously identify which one you have. Do you have the glucose side or the galactose side? And this hyphenated mass spectrometry with an orthogonal analytical method, we've used now very, very widely to analyze in particular these enzymatic reactions. And very recently at Pallister, a PhD student of mine has been working at A-Star in Singapore with the group, the bioanalytics group there, and really developed this further. So they do a lot of helic-based analysis of N-glycans, for example, where they cleave the N-glycans of proteins, biopharmaceuticals, and so on, then label them, and then actually separate them and identify them by helic. Chromatography is a very well-established system, as you know, this is sold by Waters. And they find actually that they can't always unambiguously assign the peaks because of overlap in masses. So they were looking for sort of an orthogonal technique, and so they incorporated ion mobility into their workflows. But the problem is for ion mobility you need a reference. You need references. So for each peak you would have to generate a reference, and that is actually quite challenging if you have all these different labels as well, at your fragment. So the fragment-based approach, where you take these analytes with different R groups here, depending on the label, and these might even be glycopeptides, and fragment them, and then analyze the fragments by ion mobility and determine their three-dimensional structure, and then use computation methods to put them together, is an extremely useful and much more generic way, and requires far less reference compounds. And what they have managed to do is now really take these N-glycans and resolve a lot of the stereochemical questions. So for example, when you analyze these type of structures by helic, you never quite know whether galactose is on the right hand or the left hand arm, so the 3' or the 6' arm of this N-glycan. And with ion mobility, you can separate these and very easily show which one of the two you have, which was really not possible before. So adding ion mobility to these analytical workflows is extremely useful in really getting a much better accuracy in the glycans we identify. And just to show you another example, and also actually another example of substrate promiscuity of an enzyme, we're talking here about silaltransferases again. So what Ed Pallister again wanted to do is he wanted to put as many sialic acids onto this biopharmaceutical called alpha-1 antitrypsin, which is an elastase inhibitor, because it was known that the lifetime was much increased by doing sialylation. So he galactosylated the protein that had been produced through fermentation and then treated it with various silaltransferases just to increase the silylation. And what he then actually found is when he counted the silylation very carefully is that he got much higher silylation than he was expected from the structures they had identified by Hillig. And to cut a very long story short, actually in the end he found that he can actually observe this very unusual disilylation structure, which had actually been described in the literature before as a glycan, but never been shown actually to be on a glycoprotein itself. And this disilylated structure, you could actually postulate it to be several structures. So two postulates are just shown here. So it could either be the second sialic acid could be actually on the penultimate N-acetylglucosamine. This is actually a known structure here, but there's a dedicated silaltransferase which does that. Or it could be what we proposed, this disilylated structure. We've got sialic acid on the 3 and the 6 arm here. And again, by very careful comparison to standards, you could show with this fragment-based approach using then ion mobility after fragmentation that he very, very clearly had this structure here rather than that structure on his proteins. Actually, none of this structure could actually be detected. So a very, very nice method to ask these very, very specific and detailed structural studies. I'm hoping I show you that this toolbox is ever increasing, that these can be integrated into synthetic protocols at the beginning or end of the old protocol. And I think what is the next step? I think the next step is really that we're trying to automate the whole process a little bit more and build databases that help people to identify enzymes that have actually desirable activities. So if they want to make a particular glycan, can they actually in a database find enzyme activities that help them? And we've done this recently for general biocatalysis by developing this program here, but we haven't really applied that to glycoenzymes. So that is really sort of very much the next step. So let me just thank the people. So my group, in particular, I think they've talked quite a lot about Pallister stuff. So he's just finished and is working for AstraZeneca now. My colleagues in the mass spec field and also in the, I've already mentioned Bruno Linklau and Martin Fassione and Carmen on our fluorinated sugar project. I have to thank the funders for supporting us, and I thank you for your attention.